Howdy, today we'll be going over the innate immune system. My name is Itzela Barbosa and I'm presenting with Brianda Resendez. So the innate immune system is an immediate action and non-specific response that is not enhanced when exposure to a certain antigen is repeated, meaning the innate system has no memory and will return to baseline. However, there are two main reactions involved with the innate immune system, inflammation and antiviral, and while this system responds in the same way with reinfection, it is able to recognize structures and microbes that would not be considered normal within the host cell. The innate system will recognize molecules that have breached the epithelium and through the pathogen-associated molecular patterns, or PAMPs, that have connected to a pattern recognition receptor, PRR. The PRRs are proteins that will recognize the DAMPs in PAMPs, DAMPs being molecules secreted by damaged cells, to elicit signals that activate microbial and inflammatory responses to contain or kill infectious substances. An important PRR is the toll-like receptors, or TLR, which can activate transcription factors to stimulate genes, enzymes, and proteins that can activate cells, such as the phagocytes. Once an infection agent has invaded the tissues, the phagocytes, namely neutrophils and macrophages, will respond to the microbes in order to ingest them and then fix damaged tissues. The neutrophils, which are polymorphonuclear lymphocytes, are the first to respond to infections and are the most numerous cell type. They first move to the areas of the infection and will ingest the microbes as soon as they invade the tissues. This process is called phagocytosis. However, neutrophils only function for approximately 1 to 2 days before completing their life cycle in the immune system. Monocytes and macrophages, mononuclear cells, also detect pathogens and can present antigens to T cells in order to finish with inflammation and wound healing. They are similar to neutrophils as they also ingest microbes in order to destroy them. However, they are not as numerous, and they live far longer than neutrophils do, but they do conduct phagocytosis on invading microbes. Dendritic cells also play an important role in the innate system, as they are a cell that processes and presents the antigens to T cells. However, that is as far as their functions go, since they mainly alert and do not clear invading microbes. For example, the follicular dendritic cell can be found in the lymph nodes and activates B cells and develops the memory B cells too. The way that the dendritic cells will present antigens to T cells is that dendritic cells will capture the microbe as it enters the skin, passes through the lymphatic vessels and into the lymph nodes to present these antigens to the T cells. 3 cells that are common in having cytoplasmic granules filled with inflammatory and antimicrobial mediators, as well as protecting against any hemorrhage or reaction causing allergies, are the mast cells, basophils, and eosinophils. Mast cells are activated by microbial products as they bind to TLRs and use vasoactive amines, or histamines, to cause vasodilation, to grow capillary permeability, to kill bacteria and microbes. They are also able to secrete lipid mediators and cytokines, TNF, to initiate inflammation. Basophils, not always present in tissues, are sent to inflamed areas and can synthesize mediators similar to the mast cells due to the granules that can bind basic dyes. However, the most unknown aspect of these cells is their ability to defend against allergic reactions. Similarly, eosinophils are also blood granulocytes that use their granules with deadly enzymes to damage the walls of parasites and on some occasions damage host tissue. Most of these cells stay in the peripheral tissues and increase in the blood flow when inflammation occurs.
Natural killer cells recognize any cells that are stressed by responding to them and activating cells such as macrophages with interferon Y. The natural killer cell will activate when an infected cell produces ligands to activate numerous receptors, and as the natural killer cell binds to it, it will activate and kill the cell through apoptosis. The natural killer cell will also respond to IL-12 and activate macrophages through interferon Y, so the macrophages can kill the phagocytosed microbes through phagocytosis. Unlike the similarities natural killer cells have with cytotoxic T cells, natural killer cells can recognize if a host cell is normal by attaching to the MHC1 receptor with an inhibitory receptor, which would not activate the natural killer cell and not kill the normal cell. The proteins that natural killer cells and most cells will secrete are called cytokines, and these are proteins that will mediate many functions of immune cells to stimulate growth and the differentiation of lymphocytes, activate effector cells, eliminate antigens, and stimulate growth of hematopoietic cells. These cytokines are self-limited, however, and tend to be local by binding to certain membrane receptors on the cells of interest. The complement system is based off of 9 complement proteins, C1 through C9, and activate to elicit opsonization and phagocytosis. C3b covers microbes and binds microbes to phagocytes. Lysis, membrane attack complex that disturbs the permeability of the microbe cell membrane. Inflammation, C3a and C5a promote leukocyte inflammation at complement activation. It has 3 pathways in order to be activated, which is the classical pathway where antibodies will bind to microbes and antigens, the lectin pathway where mannose binding lectin binds to mannose residues on the glycoproteins of microbes, and the alternative pathway that occurs when complement proteins bind to the microbes directly. Inflammation begins in 5 phases: recognition of infectious microbes, recruitment of leukocytes, removal of the microbes, regulation of response, and resolution for repair. Normally, this occurs as an attempt to protect the host from harmful stimuli and begin from the healing process. However, if inflammation can be harmful by subsequent repairs, normally this occurs as an attempt to protect the host from harmful stimuli and begin the healing process. However, if inflammation can be harmful by subsequent repairs, inflammation such as fever are helpful, especially in inhibiting growth of temperature-sensitive pathogens and increasing the metabolism for immune response stimulation. During the antiviral state, a dendritic cell will activate an infected cell through type 1 interferons by the interferon receptor, where the interferons will produce enzymes that will inhibit protein synthesis and viral gene expression, and, most importantly, degrades the viral RNA to inhibit viral replication. The innate system will create many responses for molecules that can provide signals that can activate naive B cells and T cells. For signal 1, the antigens will attach to the antigen receptor to activate the lymphocytes. In signal 2, the innate system will respond to microbes and host cells that may have been damaged by invading microbes. These signals will stimulate the adaptive immune system and cause the lymphocytes to proliferate and differentiate into activated cells.